Okay folks, so today we're going to look at the structure of a flowering plant. In our books, or in my version of the book, it is on page 153. You will also see at the top of page 153 that there is a QR code, so maybe when we're done you might like to have a look at that for yourself. So before we start, if you can get your hands on the following, so go out into your garden, pick a branch, obviously not ginormous, size the width of a pen, maybe the length of a pen, a little bit longer than that. That might be helpful when we start talking about some things. And if you can run to your kitchen and find yourself a carrot, a potato, an onion or a spring onion, then that may also help you when we start talking about certain things. So because you have done the new junior cycle, plants aren't really a thing, but you come preloaded with information from primary school and you should be able to name certain parts of a flowering plant. The one that you may not naturally come to mind is the transport system, but you should have all of the others, so things like roots, stems, leaves, flowers, fruits and seeds. So that's going to go into the top of page 153. Now you have to, in the bottom of page 153, draw a flowering plant. This is the posh version of the picture. I'm going to spring forward and I'm going to show you my version. I'm not going to draw it for you, but you can essentially divide it into two portions: an overground shoot system and an underground root system. So this is the posh version, and you can come back to this if you so wish, and this is my version. Now you will never have to draw this for the SEC, but it is good to have an idea of where some of the things that we're going to talk about are. So the things that are obvious are obviously the flower, the stem, the leaves, the roots. They're obvious things. But things like your apical bud, and you may see it called in a question something like terminal bud, maybe in here. This lateral or auxiliary bud, which are these little side shoots. So for anyone that's ever grown tomatoes, you have to pick out your lateral buds to ensure your tomato plant grows upwards. When we did our practical, we talked about nodes, and nodes are where the leaf comes out of your stem. And we also talked about internodes, and that's just the gap between two nodes. A new word that we have is petiole, and this is really just a posh word for stalk. In our underground system then, we will have the primary root or the main root, and you will have lateral roots or small roots that come off these. What isn't drawn in the posh picture in the slide before is this little section here, and I just like for you to put it in. So this is just showing you this is a seed, and it's obviously germinating, and when it germinates it puts down a radical which becomes the root, and it becomes a plumule which will become your shoot over time. So if you need to pause now and go to posh image or this image, and take the time to put them into your book, please. Alrighty, so over the page, on page 154 in my book, you have 3 different types of roots listed, and you have 2 diagrams on the right hand side. So right now, if you have your carrot and your onion, this is where they come into themselves. Now you may have posh carrots which have already been kind of pre-washed and slightly cleaned, so you may not, some of the roots on this may not be obvious. So tap roots, you find them in carrots, and essentially it is one main root growing from your radical, which we saw in our previous picture, and you will have lateral or secondary roots emerging from your primary root. So the main identification for your taproot is the fact that there's one main root growing from your radical and secondary roots emerge. So these are the secondary roots over here. So if you, as I say, have posh carrots, you might be able to see it, but if you have any of the little hairy bits still on your carrot, then they are your secondary roots, and this here is your primary root. And you find taproots in things called dicots or dicotyledonous plants, and we'll come up to that in 2 slides times. So carrot, dandelion, these have taproots, long roots that grow deep into your soil, can be very difficult to get up if it's a weed, and if it's a carrot you have to go down low into your soil to remove it from there. The next root is what are called fibrous roots. So you find these in spring onions, and here the radical dies away, leaving loads of equal sized roots, and these roots will come from your stem base. These will be found in things called monocots or monocotyledonous plants, so things like grass, daffodils. So when you pull them up, they've got really short roots, but all of the roots are going to be the same size. And the third, okay, so I decided to put this in here now. So we're going to have to skip, please, if you don't mind, to page 161, just to talk about cotyledons. So we, or at least I, drew these diagrams when we did our practical, these guys over here. And what we're talking about is when the seed germinates. A cotyledon is a leaf in a seed specialized for food storage. It is the first leaves that your plant produce, and this is what they look like in real life. This over here is the shell of your leaf, of your seed, apologies, that is still attached. So here in a monocot you've got one seed leaf, and in a dicot you have 2 seed leaves that are going to form. So this is the space that you have in page 161, and if you're monocotyledonous, you've got 1 seed leaf, and if you're dicotyledonous, you have 2 seed leaves. And note how I have my taproot over here, and I have my fibrous root over here. So when you're ready, we just skip back to page 154. So what I should say is that when we talk about some structures in plants, we will always refer to them being found in monocots or dicots, and eventually, as you can see on page 162, there it will be a list that we will fill in of characteristics that are specific for monocot or for dicot. So the sooner you can get them into your head, the better. So the third type of root, and it's not really a root in the true sense, is called adventitious roots, and these guys don't develop from your radicle. So the most common one, or the one you probably would be most familiar with, is if you've ever tried to take ivy off anything, there are all these little roots that make it attach to trees, to graves, whatever it is you're trying to remove it from. Also, the roots in the base of onions come under adventitious roots. In reality, these are used by a plant if there was some type of flooding and it now needs to be its own roots can't work, the adventitious roots kick in, and you can see for the ivy that it isn't a root in the true sense of the word, as in it is not absorbing water and minerals at all. So in terms of the root functions, you should be able to come up with at least 3 yourself, probably not asked at higher level, but you still have to be able to answer it if it is. So I've underlined the words that are most important here. You don't need to fill in the blurb. So they do anchorage, they absorb water and minerals. So at this point, I would say when we talk about roots and we talk about absorption, water and minerals always go together in your head, because it will help answer a couple of different questions. So they do anchorage, they do absorption of water and minerals, they do transport, and they also do food storage. So carrots, turnips, radishes, they are what are called modified roots, and they are in this case modified for food storage. Alrighty, so moving along, bottom of page 154, we have a diagram that looks like this. We have the headers for the table that are going to match these, and I'm just going to go through them now. You don't need to do anything for a second, and I'll show you what it is that I want you to do in terms of the linking of the diagram to the headers in the table. So this is a longitudinal section, so I'm cutting it from top to bottom as opposed to a transverse, which we did in our practical where we cut straight through. So in our longitudinal section we have 4 root zones. So the first zone is called the zone of differentiation, and the only thing that I would put in here is these guys are supposed to be your root hairs. The next zone is the zone of elongation. We have the meristematic zone, or can be called a cell production zone. I like to use this word because the SEC are going to ask you in questions. And the last zone is essentially the bit up here, and this is called the zone of protection. So I'm going to just show you how to link the headers to the diagram that you have on page 154. So this is what I suggest that you do. You color code them for yourself. So the zone of differentiation is the pink, and these are your root hairs. We have the zone of elongation, we have the meristematic zone, and the zone of protection is the blue. Alrighty, so we're now filling in the boxes. So in your zone of differentiation, in this zone, cells essentially haven't decided, or this is where they're going to decide what they're going to become. So kind of like stem cells at this point. So cells can develop into the 3 tissue types that we've done. So dermal for protection, ground for strength, photosynthesis or storage, and vascular tissue for transport of water, minerals, and glucose. So our next zone is our zone of elongation. Kind of does what it says in the tin. Here the cells either grow longer or they increase in size. And to do this they have to be stimulated by things that are called plant growth regulators. They're equivalent to hormones in us, and the most famous one, and the one we'll talk about in a couple of chapters time, is auxin. The next zone is the meristematic zone. And here, so meristems do mitosis. So they both begin with M's, but meristematic and mitosis go together. And here's where new cells are actively being made by mitosis. Now, just other little pieces of information that I want to put into that little column in your table is the idea that meristems are where plants actively grow. If you remove the meristem, then your plant is not going to grow anymore in terms of height or width. And the other thing that I just want to highlight for you is that we have things that are called apical meristems, and these are found at root and shoot tips. So here's where the mitosis is actively occurring, and when that happens your plant is getting longer, and so they are usually found at root tips and shoot tips. And you also have things called lateral meristems, so they will be found around the edges. So here is a lateral meristem, and again this is a tomato plant. They're always really good because you can see them. So what they will do is they will make your plant bushy. So if you're a commercial grower and you don't want a very tall plant, you would remove your apical meristem, and then your plant will, the lateral meristems kick in and your plants grow wide. And if you're a tomato grower, you always remove these guys so that your tomato plants will want it to grow up. So they are very important for commercial growers. So the last zone we have is then the zone of protection, and in your zone of protection it does again what it says in the tin. It produces a root cap, and if you think about it, this is where the plant is growing downward into your soil. So if you have a real stony soil or real compact soil, you can damage these cells here, and if that happens then your roots can't grow down. So the whole idea of the root cap is it's going to protect these root cells as they move down through your soil. Moving on to page 155 then. So we have 2 types of plants. You're either a woody plant or a herbaceous plant. And the herbaceous plant is the one that we used for our mandatory practical because we wanted to be able to cut thin sections of plant so that the light from the microscope could get through it. So woody plants are so called because they contain wood, and really lignin is the word that I want you to associate with wood here. And you tell the difference between woody plants and herbaceous because they're usually very hard and brown, whereas herbaceous plants will be soft and green and they don't contain lignin. Now on page 155 you have a table. So you hopefully will have a stem in front of you, and this will help you. So we have different parts to our stem. So this is your apical bud on this particular stem. So this is where mitosis is happening. This is where your stem is going to get longer. So this is what you're going to put into the row for terminal or apical bud. You then have these guys here at the side. So these are your auxiliary buds or lateral buds. So there should be you here, apologies. And this is where you grow side branches. So if you were to snip this off on a tree, these side guys now will kick in and your plant will grow wider. So this is what's going to go into the row of your table. And down on your stem, if you have a look, you have these little, they look like little dots. These are actually holes in your stem. They're called lenticels, and they are necessary for gas exchange. So we're in winter now. Our plant isn't photosynthesizing, it's not making food, it's still alive, and it's going to use its food stores to help keep it alive, and it has to remove carbon dioxide and water vapor for gas exchange, and it does it using these lenticels. When we do leaves, you will see that the bottom of leaves have openings in them, the stoma, and that's how a photosynthesizing plant does gas exchange much more quickly and rapidly. So lenticels now is definitely a new thing for you. So you also have this little picture on page 155 and other things that we've talked about already, but we'll go through it again. So nodes are where your leaves emerge on your stem. The internode is between two nodes, and that's the section we used for the mandatory practical. We have an axil, and that's just the angle between your leaf and your stem. This is probably more an FYI point than anything, but I'm going to leave it in there. Petioles are important. So if you can see from the picture, you probably have another word from looking at the picture here for what a petiole is. So it is just the stalk of the leaves. And there are some plants that don't come with petioles at all, and they are called sessile. So ferns are an example of sessile leaves where the leaf does not have a stalk. And a lot of monocots are also sessile. So the last little bits, and again you have this picture here in your book, and this is my labeling of it. If you want to put the lateral in here, you can too. So we have a leaf scar, and that shows you where the leaf has fallen in the autumn. So if you're looking at your branch now, that should be really clear. You see these little circular, looks like little tiny rings at certain lengths. They are called scale scar. What that shows you is the location of the apical bud in the previous year. So the distance between two sets of scale scars is showing you 1 year's growth. So again, if you have a branch in front of you, it's really, really obvious. And obviously these little dots here are all the lenticels for gas exchange. So again, you at ordinary level would have to have stem functions. You have to come up with them if they will be asked at higher level. So the obvious things, and again it's the underlined words you're looking for here: support, transport of water and minerals, transport of food, photosynthesis. So any part of your plant that is green, photosynthesis is a function of it. And finally food storage, and we'll come to that again when we talk about modified stems later on. Okay, so we are flipping over the page, and just in terms of our leaf parts, so the leaf blade is the actual leaf. we have a thing called a midrib, so that's this little section running up here. So the midrib is just a continuation of the petiole through your leaf blade, and we have the veins on the leaf that come from your midrib. Now you have the blurb for this is going to go into that little section of your table. So the veins emerge from the midrib and they contain your vascular tissue, so your xylem and your phloem. There is two types of venation that you would find in a leaf, and they look like this. And again, here's where now we look at monocots and dicots because they have specific venation. So we have what's called net or reticulate venation, and the SEC will accept any one of them. So here in a net venation, your veins form a branching network all over your leaf, and this is a trait of dicots. Whereas over here you have what is called parallel venation, so the veins run alongside each other, and this is what you find in monocots. So grasses, daffodils, cereals, these are all monocots, all have parallel venation. So in terms of your function of your leaf then, there should be some obvious things straight away that you know. And again, it's the underlined words, the important words. So if it's green, it'll photosynthesize. It loses water by transpiration, so we'll talk about that. It does gas exchange with the atmosphere. And you have modified leaves for food storage. So that is also a function. In terms of flowers, their function is sexual reproduction, and that is we're gonna hit that when we get to sixth form. So top of page 157, this is our last little bit of this. So meristematic tissue, what are you saying in your head? So meristems do mitosis, and this is where your plant is actively growing. When meristems produce new cells, they are what are called undifferentiated. So that means they haven't decided what they're going to become. So that's our zone of differentiation where they became what things? They became three things that we did in Form 4, and we've talked about them when we had to draw our diagram for our mandatory practical. We did this lovely diagram. So this is the table or version of it that you have in your book. So this is correct. This is correct in your book. This may not be. So can you please make sure you've changed the last column to what is its job? Alrighty, so in terms of our tissues, so this is going back to Form 4 diversity of cells. So we had dermal tissue, and in the little box where it says dermal, you just write in for me that this includes the root hairs, which are responsible for absorption of water and minerals. So you already know that dermal tissue is found the outside layer of your plant, and it is called an epidermis, and its job is first and foremost protection, but there is also waxy cuticle can be present in some plants for prevention of water loss. So usually the leaves with waxy cuticles are really shiny. Ivy would be an example of a leaf that has a waxy cuticle. So the other thing that they can become are ground tissue. So ground is the poor relation, the one everyone forgets what its function is, but it's found between dermal and vascular, and its jobs include photosynthesis, storage, and strength. The last tissue, plant tissue that we're interested in, is vascular tissue, and vascular tissue is found in vascular bundles, which consist of xylem and phloem. And the main job of vascular tissue, or the only job, is transport. So water, minerals, glucose can be moved throughout your plant. So we have to come out of our PowerPoint now for a second to draw the diagram, and I'm going to draw it with you on the bottom of page 157. You don't have to use the colours I've used, but I would suggest having some type of highlighter for once we've done our diagram to highlight the parts that are dermal tissue, the parts that are ground tissue, and the parts that are vascular tissue. So I need to come out of this. Discard. Alrighty, so we are drawing, it's called a longitudinal section of a leaf. And there is not even a great diagram in your biology textbook, but it's important that you are going to be able to draw it. So I am going to stay in green. So the first thing that we're going to draw is this solid box at the top. So this is the top of our leaf, the part of the leaf that will be facing the sun. We are going to draw little small rectangles. I'm just going to go this way to do it. Like so. And we are going to give each of them a swimming pool and a chair, as my second years call it. Okay, obviously the swimming pool is a vacuole and the chair is a nucleus. We're then going to draw some more rectangles, but we're going to change the shape of them. So we're going to make this so these rectangles are now drawn long ways. So again, they get so we now have pool and chair. And now we are going to draw circular cells, so they still get their pool and their chair. So I'm going to change what I draw for this one, and I'm now going to put just a whole load of little circles. This is going to be our vascular tissue, and we will color them in slightly differently when we go to our labeling. So the whole point of the fact that these cells are circular is that there are now gaps between them, so essentially air spaces. And this is important because we're going to need oxygen to be getting into these cells, and we're going to need carbon dioxide and water vapor to be released. So down at the bottom we are going to draw our rectangles again, but we're going to leave gaps. So again, they get their pool and their chair. And we're just going to give a little nucleus in here. So now we are going to go at our labeling. So up here is the cuticle. This is the top of the leaf. Here we have the upper epidermis. So this is all dermal tissue. Down at the bottom we have our lower epidermis. So this is the bottom of the leaf, the part that isn't facing toward the sun. And on our lower epidermis we have gaps, which are called stoma, singular. And these guys here at either side are called guard cells. And they will open and close to allow gas exchange or transpiration to occur. We have this tall layer up here. These are called palisade cells or palisade layer. So I'm showing my age now, but there used to be kind of a soap opera thing when I was small called Pacific Palisades. If you've ever gone to Malibu, that's where that comes from. There's all these really tall, beautiful buildings on the beach. So that's your palisade layer. This, because it has the holes, is called your spongy. It's a spongy layer, but it's called a spongy mesophyll. Technically it's a palisade mesophyll, but spongy mesophyll or spongy layer. Sorry, I didn't put layer up here. We have our air spaces, and this here is our vascular bundle. Now if you want, you can cover, I do it with X's, so then I know that this part is my xylem on top here and the phloem is going to be at the bottom. So this is your xylem and this is your phloem. So the only thing now that is left to do in all of this is to color in the different tissues. So I am going to change to highlighter. We're going to go red up here. So up here is my dermal tissue. So I hope you can see that now. So cuticle is dermal, upper epidermis is dermal, my lower epidermis is dermal. Apologies, we have to find out now what happens when we do this. Does it take it all? Nope. So we are going to color in our stoma and our guard cells. These are all part of the dermal tissue. The next one we're going to do is going to be our vascular bundle over here. So that is going to be our vascular tissue. So the xylem and the phloem, and the last but by no means least, I'm going to make it yellow so you can see it, is the ground tissue. So the ground tissue does photosynthesis, it does storage, it does strength. So the palisade layer is where you're going to get the most photosynthesis, those very tall cells, but as we've said, anything that is green is capable of photosynthesis. So these guys are your ground tissue. So if you can pause this, but I'm going to go back to the PowerPoint because in the PowerPoint I have this picture drawn again for you. Take a minute to find it, which is here. So this is just a different version of it for you. Okay, so it's showing the same things that are there with the color codes that match those particular colors. But as long as you have them matched, it doesn't matter. So this is going to end here. There is a Microsoft form that has your homework. My suggestions would be you have to learn your tables. You have to know fibrous, tap, net, reticulate, parallel, venation. You have to know sessile. You have to know the functions of the different parts of your plant. You need to know the 4 zones and where they are and what they do. And you need to understand this particular diagram because they can ask you to draw it and they can ask you to label it in terms of its tissues, which if they were to do so would look like this. So this is a microscope slide layer, a scanning electron microscope, and it's going to show you your dermal. This is your ground, this is dermal, and this is your vascular. So we draw that as the little circle things in here. So that is the introduction to structure of a flowering plant.